Investigating the effect of pH on the rate of starch digestion by amylase from scienceworksheets.co.uk. Please like, subscribe and comment to support the channel. We will investigate how changing the pH will affect the rate of starch digestion by amylase. To carry out this practical, a continuous sampling technique is used. This is where a sample is withdrawn from the reaction mixture at regular intervals, e.g. every 30 seconds, and then tested for starch. Amylase is an enzyme which speeds up the breakdown of starch into sugars. Iodine solution can be used to test for the presence of starch. So when starch is present, the iodine solution turns blue-black. If starch is not present, then the iodine solution will stay red-brown. As the reaction progresses, samples will be withdrawn at 30-second intervals and tested for starch. The first samples will test positive for starch. However, later samples will test negative for starch, as it's been completely broken down. At this point, we know the reaction is complete. Method: a single drop of iodine solution is added to each of the wells in a spotting tile. A measuring cylinder is used to transfer 2 cubic centimeters of amylase into a test tube. Another measuring cylinder is used to transfer 1 cubic centimeter of pH 7 buffer solution to the same test tube. A buffer is a solution that can help maintain a constant pH. Another measuring cylinder is then used to transfer 2 cubic centimeters of starch solution into a different test tube. Both test tubes are placed into a water bath at 35 degrees C. The solutions are allowed 5 minutes to reach the temperature of 35 degrees C. Once all of the temperatures have equilibrated, the starch solution is poured into the mixture of amylase or pH buffer. A stopwatch is then started. Every 30 seconds, starting at 0 seconds, a sample is removed from the reaction mixture and a drop is placed into one of the wells which contains the iodine solution, noting any colour change. So you will see that we've labelled them as 0 seconds, 30, 60 and 90 seconds. Initially the iodine solution will turn black to indicate the starch is present. Once the iodine solution stays brown and does not turn black anymore, stop the experiment and do not withdraw any more samples from the reaction mixture. Then we will repeat the experiment but use different pH buffer solutions such as pH 5 and pH 9. Looking at the results, at pH 7, at 90 seconds the iodine remained brown, but for pH 5 and pH 9 the iodine only remained brown at 180 seconds, so the rate is faster for pH 7 and slower for pH 5 and pH 9. To calculate the rate of starch digestion, the rate of the amylase reaction is equal to 1 divided by the time taken for the colour of the iodine solution to stay brown. So we have the different pH values and the time taken, and we have done 1 over that time taken. So 1 over 90 is 0.0111, which indicates the rate of reaction. Finally, we've plotted a graph of rate of reaction against pH, and you can see the rate of reaction at pH 7 is the highest, whereas at 5 and 9 it is lower. Thank you for watching. See our revision notes, the link is in the description.